This is a video on heart structure. We're also going to look at the cardiac cycle. Then we're going to look at the differences in pressure within the chambers and the differences in pressure within our veins, capillaries, etc. We're going to look at how the heart is controlled, how its contractions are controlled by the SA and AV, Purkinje fibers and bundle of His. We're going to look at ECGs, O2 transport, including oxyhemoglobin, and comparing that with fetal hemoglobin, and then CO2 transport and the Bohr shift. This is following on from my previous video, which was looking at circulatory systems 3.2 and all this content. If you want to look at that video, you can. But first of all, let's get into heart structure. So we know there's 4 chambers. You've got left atrium, left ventricle, right atrium, right ventricle. And we've got 4 vessels, 2 veins, pulmonary vein and vena cava, so the pulmonary vein takes in oxygenated blood from the lungs into the left atrium, and vena cava takes deoxygenated blood from the body into the right atrium. And we've also got 2 arteries, the aorta, which takes oxygenated blood from the left ventricle to the body, and the pulmonary artery, which takes deoxygenated blood from the body, which has been pumped from the vena cava, from the right ventricle into the lungs. Right, that was in a nutshell what's going on here. So this is a diagram from my notes that I've taken and put in here because I thought it's quite handy to look at. It's not what the heart actually looks like, but it puts that really nice and basic for us. So one key word here is on the side is myogenic. So heart muscle is myogenic. It's not neurogenic, so it's not controlled by a nerve. It manages to contract itself, and we're going to see later how that works. So this is cardiac muscle, drawn in green around that side. As we notice in the atrium, it's much thinner. The atria are much smaller chambers. They do not need to create as much pressure, as all they're doing is pumping the blood from here into here. Okay, so you don't need to contract much at all. Then we've got our ventricles with a thick cardiac muscle. Notice the left ventricle cardiac muscle is thicker than the right, and that wasn't by accident. It's because the left ventricle needs to create much higher pressure than the right ventricle, as it's got to pump the blood a lot further. There's a lot more resistance. It's pumping it through the aorta to the rest of the body, whereas the right ventricle is only pumping it to the lungs. So the blood's going to feature much less resistance, so much less pressure is needed to be generated. Okay, so there's 3 steps here. The left atrium, right atrium, and the left ventricle and right ventricle, they work at the same time. You don't have a left atrium fill up and the right atrium fill up. They happen at the same time. Okay, so first of all, blood comes in to the left atrium and right to our atria, and it basically fills in the left atria and the left ventricle, because this atrioventricular valve, I'll just say atrium ventricle now because we shouldn't be specialising either, they both do the same thing. This atrioventricular valve is always open at this point, so blood is pooling in until there's then atrial systole or systole, so the atria contract and push all the remaining blood into the left ventricle. This causes loads of blood in here to be at a really high pressure, which closes the atrioventricular valve. So we're going to look at the valves and how they work a bit later on. Then there's a slight delay, and then we have ventricular systole. I'll go through this cardiac cycle in 1 second, which pumps blood through our arteries and to the various locations that I've already spoken about. Okay, now we've looked at that really quickly. It's not too complex. Let's look at the cardiac cycle, which is equally fairly simple. So first of all, we've got the left ventricle and the right ventricle contract from the apex to the base. Okay, that is called ventricular systole. So both the left and right ventricles contract at the same start, starting from the base, pushing the blood upwards and out. So here's the base, they contract here, the wave excitation passes up here and up there, causing the contraction. Okay, then we have a period of diastole, or distole, however you prefer to say it, when the muscular walls of all 4 chambers relax. Elastic recoil causes the chambers to increase in volume, allowing blood to flow and fill the chambers. So at diastole, we've got the blood filling the chambers. At ventricular systole, that's when the blood's being pumped out of the ventricles through our arteries. Finally, we've got our left atrium and right atrium contract together, which fills the ventricles with blood. Okay, so at this point here, during diastole or distole, we've got blood filling these chambers with this valve open, and they're pretty much one chamber while this valve's open. It's all filling up. It'll probably fill up to about this line here, and then the atria contract and push even more blood, forcing blood into the left ventricle as this valve is closed. More blood's forced into here. The left ventricle then closes the valve because the pressure gets so high in here, and the left ventricle then goes under ventricular systole, forcing blood out of the arteries. That is the cardiac cycle here, and then we can just look at how the valves work. So we've got the atrioventricular valve first, which is here on the diagram between the atrium and ventricles, and let's look at the processes as to how this valve here closes. So pressure in the ventricles rapidly drops below pressure in the atria. Blood pushes atrioventricular valves open. So this is when the blood's filling in. So the blood fills up in the ventricles, and this is naturally open, okay? Blood entering the heart pushes straight through the atria into the ventricles. As the valves are open, chambers become one. The pressure rises slowly as they fill with blood. That's pretty simple. So then the valves remain open during atrial systole. So this valve will close and push all the remaining blood into the ventricle. The ventricles fill with blood, and that pressure causes the valve to close. Okay, so pressure in here, blood pushes against the sides up here, and it forces it to close. So the blood pushes against these things here called tendinous cords, which also prevent the valve from inverting. The contraction of the ventricles forces this valve closed, allowing blood to pump out through our arteries. Then we've got semilunar valves, which are, I'll show you on the diagram. They're the valves here, okay, at point 3, going into the arteries. So semilunar valves, before contraction, the pressure in the major arteries is higher than the pressure in the ventricles. So when we've got blood in the arteries during ventricular diastole, we've got blood just sitting in our arteries here, and that pressure forces these valves closed. Okay, ventricular systole, so when the ventricles contract, this pushes these valves open. So as we can see here, the chamber here, which was large, the ventricle chamber was really large, then when it contracts, it gets really small, we've got really high pressure, and it forces these open and forces blood up through the arteries. Right, so now we're going to be looking at the different pressure in our chambers. So we've got 3 things here to look at. We've got atrial systole, ventricular systole, and diastole. They are 3 stages of the cardiac cycle that we've already looked at. If you don't understand it yet, try and recap that part of the video so you do, because you will not understand this if you don't understand the cardiac cycle. Now, atrial systole means, or systole, however you say it, I always change it, sorry. My biology teacher tells me it's systole and diastole, but I naturally say systole and distole. The atria is contracting during atrial systole. The atria contracting in this period, this means pressure is going to rise because the chamber is getting smaller, so of course pressure will rise. And it rises to this point, and at this point the AV valve closes, the atrioventricular valve closes. And let's think what happened in here. So the atria are no longer contracting as we enter ventricular systole. So the atria are relaxed. We have atrial dystole. So if they're relaxed, they're larger, pressure is going to decrease as we enter ventricular systole. It only reaches a certain point here, so here it gets at its lowest, starting to fill up with blood, because its semilunar valve, its valve from the vein is still open, so pressure begins to rise. Okay, because this line here is blood filling in the atria again, while the valve's closed. Then we get to this point here. Probably drawn that line a bit too steep, so I'm going to draw it again. We get to this point here. So at this point we're in, distole. Okay, so both the atria and ventricles are relax. They become one chamber as the atrioventricular valve opens. So now the atria and ventricle become one chamber. That means they've got a much larger space as this valve's open, so it decreases the pressure in the atria. So as we'd expect, the pressure decreases. And towards the end of this, we're going to be, I'll do it like that, and then it's going to be rising again as we go back to the start. So this is a cycle. It goes up, goes down. There's not too much difference in our atrial pressures. It's small. It's not put under serious pressure. Okay, so that was our atrial pressure, which is the easiest to explain. So if you found that simple, well done. If you didn't, unlucky, because it's getting harder now. We've got next to look at our ventricle pressure. Okay, so at this point during atrial systole, remember our AV valve is open. Okay, so at the start our AV valve is open, so they are one chamber. The atrium and ventricles are one chamber, therefore the pressure is the same. So up until the point where this closes, the atrial and ventricle pressure is exactly the same. Now we get to this point, the AV valve closes as we enter ventricular systole. Okay, and we're looking at the ventricle here in blue. So if the ventricle is contracting, pressure is going to suddenly rocket up. Remember, we've got really thick cardiac muscle around our ventricles. This is because the pressure they generate is huge. Oops, I just drew that wrong. Let's draw it to this point. So our semilunar valve at this point opens. So here the ventricles contract, the blood forces this valve open, which we spoke about just before. The pressure builds up, forces it open. The pressure continues to rise because we are still contracting until it reaches a peak. Here the pressure remains constant while we're squeezing all that blood out, and then our pressure drops slightly and our semilunar valve closes at this point. Okay, so our semilunar valve closes, and therefore we're at the end of ventricular systole. So the ventricles are now entering diastole. That means the pressure is now decreasing, and we see that go rapidly. Okay, so let's draw this line through diastole. We get to this point here, didn't join that up, that's really annoying me, where the AV valve opens. They become one chamber. The ventricle pressure is the same as the atrium pressure. So while the AV valve is open, the pressure remains the same in the two. We've got one more thing to look at on this diagram, and it's going to be our aortic pressure. And I think I will do this in orange. Okay. So first of all, we're doing a straight line across until the semilunar valve opens. That's because our pressure in the aorta is constant. The aorta is really narrow. We've got blood pooling in there, keeping this valve closed. Then we enter ventricular systole, so the ventricles contract. They become one chamber with the aorta. Okay, so the aorta and the left ventricle are now one chamber. What we've said, when a valve's open, the two chambers become one. Okay, the two chambers the valve connects become one. Therefore, their pressure remains the same as each other. Closely match that as much as I can until this valve closes. Okay, that was too steep. The pressure rises in the aorta as the valve's closed. It's a really narrow space. Took 3 attempts at that. Finally got there. So we've got to remember that this is a cycle. So once it reaches the end, we're going to go back to start, and that's the same for all three. One thing I'll add on here is what these things are, and this one is our aorta, remember. Then we've got our atrial pressure, which is in purple or pink. Atrial. And then we've got our ventricle pressure. That's the wrong colour, but you know what I'm talking about with blue there. Okay, so that is the different pressure in the different chambers. Really quite complex, but the key thing to remember is when a valve opens, the two chambers it connects, two chambers that it's connecting become one. They become one chamber. Okay, because there's nothing closing them off. They share the same pressure. So therefore, when we've got two chambers as one, they're going to have the same pressure. So when the semilunar valve opens, the aorta and the left ventricle have the same pressure. When the AV valve is open, the atria and ventricle have the same pressure. Okay. Go over that again. It's not easy to get a grip of, but it is quite simple once you understand it. And the difference in pressure is to do with our total cross-sectional area. So as you see in this diagram, the larger the cross-sectional area, so our capillaries, which have a huge cross-sectional area, we've got so many capillaries, the blood is then distributed through so many capillaries, it has so many alternatives, the pressure, which is shown in this diagram here, is much lower. So aorta, the pressure is at its highest, then it enters arteries, arterioles it significantly drops, as there's several hundred of arterioles, and then thousands, if not millions, of capillaries. So that's when we see our lowest pressure. Then it enters the venules, it rises, and the veins, and then the vena cava. Okay, there's not really too much to talk about in this diagram. I'm just going to look at my notes to make sure I don't miss anything. So tissues require blood to be delivered in an even flow. Artery walls have a lot of elastic tissue. So when blood leaves the heart, they stretch as blood moves out of the aorta. Pressure in the aorta drops. Elastic recoil helps maintain blood pressure in the aorta. As the blood flows through arteries, fluctuations become less obvious. Okay, so what this diagram's not shown to you is when blood's leaving the heart, in the aorta, we're going to be seeing this, fluctuations. They could become smaller and smaller until we can't see them at all. And these fluctuations are caused by our heartbeat, so a contraction, relaxation. Okay, that's the only thing this diagram's missing. The pressure gradient between the arteries and the arterioles causing blood to keep moving through. Okay, and we know, and we might be seeing that our pressure is going up here. We might think that's an issue because the liquids always move from high pressure to low pressure. However, we remember that our veins have specialised methods for venous return to get blood back. They've got their valves, and we've got contraction of muscles to help move blood back to the heart. Okay, so that was the different pressure in our vessels. Now we're going to look at how the heart produces a signal. So we've got 4/5 main things to look at here. So first of all, we've got the SAN, the sinoatrial node. This generates waves of electroactivity, waves of excitation. The rate at which the SAN produces these waves of excitation is controlled by parasympathetic and sympathetic nerves. So the sympathetic nerve is called the accelerans nerve. It releases neuroadrenaline as its neurotransmitter, so it's much faster. The vagus nerve uses acetylcholine that we see in muscles so to produce a slower heart rate. The pace at which it is creating these electrical activities is dictated by which signal it's receiving, whether it's sympathetic or parasympathetic, or whether it's got adrenaline, whether adrenaline is having an effect on it. Okay, so we can ignore that anyway. So the sinoatrial node sends out this wave of excitation. It causes contraction of our atria. However, it does not cause contraction of ventricles, as we have this band of non-conducting collagen fibre which prevents the ventricle contraction, as we want it to be, from the bottom to the top, from the apex. Okay. The wave of excitation is then picked up by the AVN here. It picks up the signal and passes it to the bundle of His, which I've drawn on here, which conducts the signal to the Purkinje tissue, or Purkinje fibres. They carry the wave of excitation through here, and cause the ventricles to contract and squeeze blood up through our arteries. Okay, one thing to note here is that AVN, atrioventricular node, causes a slight delay in contraction to make sure that atria and ventricles contract one after each other. That's hence why when you have a heartbeat, you take a pulse, you can feel a dun 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 dun. The first one's the atria, and the second beat you hear is the ventricles contracting. Now let's look at ECGs. There's three main things to look at. You've got the P wave, which shows excitation of atria, the QRS complex, which shows excitation of ventricles, and the T wave, which shows diastole or distal. Okay, and that's shown here. Once again, let's take it from my notes. There's some ECGs you need to learn. So we've got a normal ECG, which would look like this. Regular intervals. It's consistent. Okay, large QRS complex. Consistency. This is bradycardia. So when we've got then we've finally got tachycardia, which is a fast heart rate. So we've got lots of them produced. Two more to look at is atrial fibrillation, where the atria are beating more regular than the ventricles. So as you can see here, we've got lots of little P waves and T waves. We can't even distinguish which one's which. The atria are contracting way too often, more often than they should be in our normal heartbeat here. And then we've got ectopic heartbeat, which is an early ventricular beat. This is showing ventricular fibrillation when the ventricles are contracting way too regular. But at this point here, we see an ectopic beat that I'm circling here. It is an early ventricular contraction. So we see it contract in our QRS complex, and it contracts again. Two QRS complexes, okay? So learn these ECGs. You might want to look at some better diagrams yourself, as especially these two aren't that helpful, but I've got them in my notes, so you should probably do a bit more research into them. Now we're going to be looking at oxygen transport. We've got three stages. So when we're looking at oxygen transport, remember this takes place in red blood cells in the hemoglobin. So when oxygen binds to hemoglobin, it forms a compound called oxyhemoglobin. Hemoglobin has 4 subunits. Each heme, which contains an ion, has the capability to hold 1 O2, and we've got 4 heme groups. So each hemoglobin molecule can carry 4 oxygen molecules. Let's look at how hemoglobin is saturated. So we've got 3 stages here. We've got low partial pressure of oxygen. So for example, within the capillaries going to muscle tissue, we've got a really low partial pressure of oxygen. So little saturation of hemoglobin, lots of the oxygen dissociates as surrounding it is a low partial pressure. So heme groups at this low partial pressure are found at the center of the hemoglobin. It is really hard for oxygen to bind to the heme groups, as they're not really ready for association because they're hidden within the hemoglobin, so they're right in the center, hard to bind to. As the partial pressure increases, one oxygen associates with a heme group. This causes a conformational change of hemoglobin in hemoglobin, allowing, so that's a change in the shape of a protein when we see that word conformational change, allowing more O2 to readily associate, hence the S-shaped curve. So here, not many oxygen molecules are associating, then all of a sudden at this point, one oxygen molecule will associate, allowing the conformational change for more oxygen to associate. So this conformational change takes the heme groups away from the center, making them much more accessible, easy for oxygen to bind to. Then finally we reach 100% saturation, where O2 will readily dissociate into, after the blood has left the lungs, we usually expect 100% saturation. Okay, so that is oxygen transport. Then we're going to look at fetal hemoglobin and its differences with adult hemoglobin. So fetal hemoglobin has a much higher affinity for oxygen than adult hemoglobin, as fetal blood needs to associate at a much lower partial pressure. So adult hemoglobin is formed of 2 alpha and 2 beta hemes, whereas fetal hemoglobin has 2 alpha and 2 gamma. I think that's the Y stands for gamma. Not good on my Greek. Heme groups. So it's got a different 3D structure to our adult hemoglobin. This allows fetal hemoglobin to bind to more oxygen easier than adult hemoglobin does. So therefore, when the mother has a low partial pressure in the placenta, the fetal hemoglobin absorbs more oxygen from the surrounding fluid. So if fetal hemoglobin was the same as adult hemoglobin, there'd be no need for the oxygen to dissociate from the adult to the fetal. So fetal hemoglobin needs to have a much higher affinity for oxygen, otherwise they would not be able to access the oxygen from the mother's blood. So fetal hemoglobin absorbs more O2 from the surrounding fluid, the partial pressure of the placenta gets even lower, so maternal blood releases even more O2. Okay, so as the fetus takes up the oxygen, the partial pressure in the placenta gets lower, and even more oxygen is released into the placenta to be taken up by the fetus, as oxygen will dissociate into a lower partial pressure. So the main concept here is O2 dissociates with adult hemoglobin and associates with the fetal hemoglobin. That's that in a nutshell. You might want to do some further reading into it, especially into the formation of two types of hemoglobin here, but I think that is spoken about in detail in Unit 2. Okay, finally we're looking at carbon dioxide transport, and there's 3 ways this happens. It's 5% of it is done in the plasma, 10% in carbaminohemoglobin, and 85% in hydrogen carbonate ions. So first of all, we have this reaction here. CO2 plus water creates carbonic acid, which is this H2CO3. That happens in the red blood cell. Okay. H2CO3 then dissociates into hydrogen carbonate ions and hydrogen ions. Hydrogen carbonate ions diffuse out of the red blood cell into the plasma. This causes chloride ions to shift into the red blood cell. This is called the chloride shift. Builds up within our red blood cell, lowering the red blood cell pH, and we know the dangers of that in a protein. So this pH needs to be buffered, and the way we do that is through hemoglobin. So hemoglobin acts as a buffer to maintain constant pH. The H plus ions, so hydrogen ions, bind to the hemoglobin, forming hemoglobinic acid, HHB. Okay. For this to happen, the O2 must dissociate with the hemoglobin, so oxyhemoglobin is no more. Oxygen dissociates from the hemoglobin, allowing the hydrogen ions here to bind to the hemoglobin. So this is called the Bohr shift, which I'll draw on the graph. It's exactly the same, but lower. So it follows the same pattern, but it's lower, as the presence of hydrogen ions shift to the right of the association curve. So let's really quickly highlight the steps. Carbon dioxide and water bind within the red blood cell, forming carbonic acid. This dissociates into hydrogen carbonate ions and hydrogen ions. The hydrogen carbonate ions move out of our red blood cell, causing a change in potential, and to equalise this potential, we have chloride ions, which are also negative. They shift into the red blood cell. That's our chloride shift. The hydrogen builds up, lowering the red blood cell pH, and to buffer this lowering pH, this acidic red blood cell, the hemoglobin acts as a buffer. It binds to the hydrogen ions, causing oxygen to dissociate from the hemoglobin, and a lowered association of oxygen overall. Okay, so that is the end of Unit 3.2. I know it's quite long, but thanks for listening.